I'm Dr. John Timms and I lead the Cancer Proteomics Group within the Department of Women's Cancer. The group's major research themes are the identification of cancer biomarkers for early detection and diagnosis, and also understanding the molecular basis of cancer through the application of proteomic technologies. Proteomics is the study of the protein content, or proteome, of cells, tissues and biological fluids such as blood. It involves the identification and quantification of hundreds to thousands of proteins at a time across a set of biological samples, for example, blood specimens. We primarily use a technique called mass spectrometry, where the accurate mass and levels of the peptide components of proteins are determined, enabling their identification and relative quantification across the sample set. Mass spectrometry is linked downstream from various gel or liquid chromatography-based protein and peptide separation strategies, enabling improved coverage of the proteins in a sample. In a typical biomarker discovery experiment, we would compare serum samples from cancer-free control women to those diagnosed with cancer, looking for differentially expressed proteins as potential biomarkers. Uniquely, we are looking for early markers that will improve survival using samples taken prior to diagnosis of cancer from women recruited to UKCTOX, the world's largest ever randomised controlled trial. The next step is to validate the markers using immune-based assays and to build multi-marker models that outperform existing markers used in the clinic. We are currently validating such models for ovarian, breast, pancreatic and biliary tract cancers and hope to translate these to the clinic as diagnostic tests. Other basic cancer research has defined novel gene functions and signalling mechanisms involved in promoting breast and ovarian cancer,